Chicago Tribune.
And so we're going to go into a little bit more in depth on our collaboration, which was published last year. Sam's article was published, and my 2 scientific articles were published all last year, throughout the course of the year. This will be very informal, and the idea is to go in depth on, I think, whatever you want to talk about. So I think the idea is we'll start talking, and then if there are particular areas of interest that you want us to dive in on, then we'll go in on those. And it's really guided by what everyone in the room wants to talk about. As a starting point, I'm just going to go over some of the science, and I'm going to keep it brief and high level, but please feel free to ask questions because, of course, I love to talk about the science, and I'd be happy to go in more detail if you'd like. So my research is in observational data analysis, and that is a complex way of saying just forming hypotheses based on observations. This is not a new idea. Observation has always been the starting point of scientific discovery. Does anyone in the room recognize this figure up here?
Yes.
Yes?
Charles Darwin.
It's out of Darwin's notebook. It's a scanned page out of his notebook, and I love it because up there in the top left corner he writes, "I think," and then he draws this beautiful diagram. And you can start to see that he's relating geography and phenotype, which will of course form the hypothesis of evolution. And Darwin is not the only one who does this. We do this every day in science, and I study adverse drug events and drug-drug interactions, and so the fathers of pharmacovigilance, as we call it, or drug safety science, are William McBride and Lentz, and they observed that the patients that they were taking care of that were pregnant and were exposed to thalidomide commonly had birth defects in those babies. And they formed a hypothesis that thalidomide use was causing these birth defects, and they validated that hypothesis and really launched modern drug safety surveillance. Now we have other tools for observing. William McBride and Lentz and Darwin went out into the world or had their patients come to them, made observations with their senses and their eyes, and came up with hypotheses that way. But today we're collecting data on every patient that walks into the hospital, enormous amounts of data on those patients. The physicians they see, the drugs they take, the diagnoses that were made, the labs that were run, genome sequences for some of them, heart rates, you name it. We are collecting it and storing it. This is just the adoption rate of electronic health records across the United States. Largely, it's almost 100% across the United States now. At one site alone, at Columbia, we have over 5 million patient records. And just to give an example of the scope and scale of the size of the data that we're dealing with, we have, in the last 10 years, 30 million prescriptions have been ordered, 100 million diagnoses have been made, 500 million laboratory results have been run. Somebody's taken some blood, run a test, or taken a urine sample and run a test. And these are just 3 of the data elements of hundreds of different types of data that are collected and stored and available for research purposes. So we're going to focus on how do we use these data to come up with new hypotheses about drug-drugs interactions. The question motivating a lot of my work is that there are drug-drug interactions occurring, they're causing adverse events in patients, and we do not know that they exist because there's no other formal way to study them. This huge data set gives us a new opportunity to study these interactions by examining these data. Drug-drug interactions can occur anytime a patient takes 2 or more drugs simultaneously. They account for up to 30% of adverse drug reactions that occur. And by understanding drug-drug interactions, we can improve the outcomes for our patients. First off, if we know that 2 drugs are going to adversely interact with each other, we can simply not prescribe them together. That would lead to better safety outcomes. But also, if we study the pharmacology and the biology of how 2 drugs are interacting, that can reveal something about the disease biology that we can use to our advantage to maybe design synergistic therapies in the future. Kind of a long-term goal of our research. Clinical trials do not typically investigate drug-drug interactions. It would be very laborious to require a drug company to do that. They do avoid drug-drug interactions. They do some pharmacokinetic screening, which is a simple way to identify drug interactions, but is not very reliable. And so essentially we have to turn to these observational studies of data sets like the electronic health records to study these effects. One particular area that we're interested in are drug-drug interactions that prolong the QT interval. And this is some technical diagrams to show you what that might look like. Here, if you hook yourself up to an ECG or an EKG, you'll get this nice pattern that everyone can recognize as your heart beats. The long QT is this wave gets extended. You can see the difference between these two. And the danger of having your heart rhythm look like this on the ECG versus that is that it could develop into something called torsades de pointe, which is this turning of the points or very erratic rhythm of the heart. And when your heart is doing this, essentially the top part and the bottom part are not fibrillating in sync, and that means no blood is really getting pushed into your body. And that's what's dangerous and could be causing death.
So some people are actually born with this condition right here. You might be born like that, but there are also drugs that can cause that. And so that's one thing we're interested in. As a journalist, I was interested in this condition because it's a really mysterious and baffling condition to have. For instance, if you were to die of a heart attack or someone just found you dead, the doctor may conclude that you were old or it was your time to go, when it was actually this condition. Or often if you're a very good swimmer and you drown, people are baffled by how that could happen. He was such a good swimmer, why would he die? Maybe he was drunk. No, it could be this. It could be this condition. So as a journalist, I really love this condition because it was affecting people, but it was also mysterious. So it was interesting to work on.
And because of that, because of the high risk, there is actually a very simple test to determine whether or not a drug is likely to cause this. And it's called, it's a test on the hERG channel. It's a very important channel in the heart. All drugs that are known to cause this syndrome do it by blocking the hERG channel. And so the FDA requires, since the mid-90s, right, 94, that every drug be tested against the hERG channel before it could get approved. And if you show any activity, if that drug is affecting that channel at all, it essentially kills the drug. Only in rare cases where you really have a reason to prescribe that drug, maybe it's an orphan disease or something like that, would the FDA consider allowing it to move forward. Drug-drug interactions have never been systematically tested this way. And in fact, before our finding, there's really only one known drug interaction that caused this, even though we suspect there's more. In order to identify these drug-drug interactions, we had to invent a new data mining algorithm. Because the data, nobody's reporting a couple of drugs and long QT syndrome to the FDA and making it really simple for us. What they're doing is they're reporting all types of symptoms with all types of different drugs. The data are really noisy and mixed together. And so instead of using a traditional approach, which the FDA would use, we had to invent something new. And it's based on a very simple idea. It's that it's based on the idea of how physicians or doctors diagnose disease. So when you come into the clinic and you have a disease, let's say diabetes, that disease isn't written across your forehead. It's lying under some level of detection. You can't just see the diabetes. But you can measure certain effects of having diabetes. You can measure high blood glucose, or you have pain in the extremities, or some fatigue. And that collection of observable effects allows you to inform the hypothesis of the disease underneath. So you say, oh, I measured all of these effects, like your blood glucose, and now I hypothesize that your disease is diabetes, and I'm going to treat that disease. So we're going to do exactly the same thing, but we're going to do it with adverse drug reactions, drug-drug interactions. We're going to have some unmeasured severe effect. That's going to be prolonged QT syndrome, acquired long QT syndrome. And our hypothesis is there are other more commonly reported effects that are going to go along with it. The collection of reporting of those effects, it will be indicative of the presence of that underlying or not reported long QT syndrome. So that's exactly what we did. We built one of these profiles. These are 3 of 11 different features which are in this profile, and these are just a couple of them. And there are other types of arrhythmias, and so how they're linked to long QT syndrome makes some sense.
So if I could stop you there, because when you first explained this work to me when we got together, it was almost like how astronomers can discover and infer the presence of black holes. An astronomer might not be able to see a black hole directly, but you can infer the presence of a black hole by the effects around it, like the gravitational pull on neighboring stars and things like that. Is that right? It's sort of similar. You're looking for that fingerprint or the other effects.
That's exactly it. So this long QT syndrome is like the black hole, and its pool or its association with these other effects are what you can observe. And so you're looking for those to infer the presence of the thing you can't see.
So as a journalist, I was thinking in my head, well, he's making the invisible visible. I really like that. And when I came back to my bosses, there's a lot of technical stuff here, and the bosses, their face would go blank, right? I said, well, he's making the invisible visible, and they would like light up. You know, it's like, okay, this is cool. So it's cool work, but you had to like find a way to explain it.
Yeah. So we applied this algorithm, we mined these large data sets, and we came up with one prediction over. Yeah.
You say you link these data sets. It means that these three different types of measurements, you see, is it connected to one single patient, or is it just you?
No.
It's not.
It's not necessarily connected to one single patient.
So the three things could be from different patients.
Absolutely.
You just collect a number of these different measures.
Yeah, we use something called a supervised machine learning algorithm to identify a profile of these effects. And so that's how we came up with them. And it collapses the data, and it integrates it all across all the patients that we have. And so the prediction that kept coming up in all of the data sets that we tested was between ceftriaxone, which is a common antibiotic. In our hospital, it's a first-line inpatient antibiotic, and lansoprazole, which is a proton pump inhibitor used around the world. It's available over the counter in the U.S. as Prevacid, and it might have a different name here, but it's most certainly available. It's very common and very likely that patients could be on both of these drugs simultaneously, especially if they are taking lansoprazole at home, they come into the hospital, they get put on an antibiotic, and this reaction could start.
In fact, my child was on this lansoprazole for a number of years, and it helped to sell the story to my bosses again when I could go across the street in Chicago to the pharmacy and come back and say, here's the drug we're testing, because you can buy it off. And so you're trying to take the science and go back to the newsroom and try to get them to understand quickly, because when you meet the bosses and discuss this, you have a few minutes to make your case. And so anytime you have a prop, right, you're dealing with it and say, here, this is what we're testing. I just bought it over here. This could potentially be harmful if taken with this other drug.
Sorry, one little bit. What is it then? Like, so it's the first one's an antibiotic and the second one's a...
Proton pump inhibitor?
Yeah, I know it says that.
So
acid reflux.
Acid reflux drug? Yeah.
So a lot of children, they throw out food as a young person because of throat blisters.
Yeah. It's a, for multiple types of kind of gastrointestinal distress. It's used for a wide range of things. It was originally approved for GERD, but okay. So at this point, we had a lot of data from observational sources that suggested that this interaction was happening. But that's not going to convince any other scientists. They're not going to believe that these two drugs are causing the effect. They're going to say it's associated with the effect, that's what you found. But it could be because patients on two drugs are sicker than patients only taking one drug. Maybe that would explain your effect. Or they'll say patients who are on ceftriaxone are worse off than patients taking other antibiotics, cephalosporin antibiotics, and maybe that'll explain your effect. There's always another reason that a smart epidemiologist will come up with to invalidate your finding. So you have to do some sort of prospective experiment, and you have to do something that the community is going to trust. And so what we did is we did the experiment that the FDA requires of every drug, and we tested these two drugs in combination now against that hERG channel, that really important channel in the heart. And this is the result of this experiment, which is really kind of a cool experiment. So you trap a single cell on a glass plate. So you have this glass plate, and it has one little tiny hole in it. And the hole is so small that only one cell can fall into that hole, like a billiard ball.
A hamster cell.
It's a
hamster
hex
cell.
cell.
Okay.
Yeah. Is it a no, it's a human cell.
Okay.
Or are they
I thought it was a hamster cell.
Maybe it is a
Yeah,
hamster
okay.
cell. Yeah, maybe it is. Maybe I've been saying that wrong. But anyway, it doesn't really matter because it has the human protein expressed on the outside. And so it falls into this hole, and then you suck really hard with a vacuum on the other side, and it blows out the cellular membrane. Remember, we have this membrane keeping all the insides of the cell inside the cell. So it blows it out, but it keeps it stuck in that hole. So now you have the inside of the cell exposed to one side of this glass plate, and then the outside exposed to another. And you can do this experiment where you run current, a voltage between those two, and measure the current. And that current is the activity of the herd channel. And that current is what's being plotted here. So we haven't really, this is like nothing, this is baseline. You invert, there's some technical details that aren't really important. You invert the current here, and then you turn it back off, and then that's when the herd channel releases. And so the activity of a herd channel is represented by this spike. Okay, so here in the top, we did one drug alone, ceftriaxone alone. Now both these drugs have been tested against the herd channel before, so we know what we're gonna get. And so this is exactly what you see. We increase the concentration as the green color gets darker, and no change happens. The spike goes in the same spot every time on the same cell. Now, if we put ceftriaxone in the presence of lansoprazole, so now the drugs are together, you can see when it's low lansoprazole, it spikes and goes to the same spot, and then as low ceftriaxone, spikes and goes to the same spot. As we increase the ceftriaxone, the activity gets reduced and reduced. And then if we increase the concentration of lansoprazole, you see the same thing, even more striking. This is just the vehicle, and then this is in ceftriaxone at increasing concentration. So you can see this really striking dose-dependent response. And that's exactly what you need to show to prove to other scientists that the effect is real. And this is that experiment that I was really nervous about, because there are 100 reasons that I could name why this wouldn't work. That doesn't mean that the drugs aren't interacting. This could still be real. But there's 100 reasons why this experiment could just not work and not show what we want. But only one reason why it would. And the one reason why it would is because these two drugs are interacting and affecting the herd channel.
It was a lengthy process to do the cell research because you had to get healthy cells, right? A lot of times we were waiting to make sure the cell was perfect. It had to be a perfect cell in order to test it.
Yeah.
So the cell testing, in terms of doing the research, it took almost a year, didn't it? But
cultivating the cells is very
If you hadn't done, could you have published a paper without doing the cell testing, you think?
Not with the same impact.
Right.
Not even
the same
close.
thing. Yeah.
It would be a paper that would be almost completely ignored. And you know about the follow-up, the Italian follow-up, right?
Right.
Well, I'll just finish this real quick.
Okay.
But then we can talk about that story because that's really cool as like the last thing. And so what we did one more thing, just to prove that the data science and the data mining is doing what we want it to do. We went back to the algorithms and we said, you did a really good job at predicting one positive. Now give me one predicted negative, drugs that are not going to interact, right? Because if we can do one, we should be able to predict the other. So that's what we did, and we made them be the same class. So we have cefuroxime is another cephalosporin antibiotic. You know they're similar because they're spelled similarly. And also lansoprazole. But this time, the algorithms are telling us there is no expected effect. And so we took that into the lab, and we measured it in without lansoprazole and with lansoprazole, and we found no difference in the effect. It's exactly right. Which is really went a long way to convincing the reviewers at this journal, which is a very prestigious cardiology journal. It's one of the top, it's either the top one or the second one, depending on who you talk to. And it went a long way to convincing them that what we were doing was actually valid. And the really cool thing is, in response to that, because this ended up getting a lot of scientific press, in response to that, we had a letter to the editor written by this Italian team. And the most striking, they presented 2 additional pieces of evidence in support. Anytime you get a letter to the editor in response to your paper, you're like, oh, but this was really, really interesting. And they had 2 different pieces of evidence, but I'll only say the one. And the one was they had a patient show up. They put them on ceftriaxone. And just like I was saying, they were on lansoprazole. They came in, they put them on ceftriaxone. And their QT, they were monitoring their QT, and it went way up. Then they took them off lansoprazole and switched it for a different proton pump inhibitor, and the QT normalized. Which is, again, really strong evidence that this is a causative effect, ceftriaxone and lansoprazole. That's my last slide. That's the science.
It's pretty cool, right? I mean, what I liked about it is that we started with a billion possibilities, right? The universe of big data, all billions and billions of bits of information in your archive, and all these possibility drug combinations. And after all this work, we're down to a single cell, right? And testing a single cell. And so that day, the photographer was there. We were all in the room. And yeah, it was sort of, it was nervous, right? Because if it worked, you knew that the project, as a piece of journalism and science, would be here. And if it didn't work, maybe it'd be down here, or maybe we'd keep going. But I think at that point, we wanted to at some point, you can't do this for 30 years, right? I mean, we had to end it at some point. So it was an exciting moment.
Yeah. And in fact, the experiment started working the first day we tested it, and it was showing the effect. I mean, it's a very robust effect. We've done this experiment hundreds of times now, essentially, and it's a very robust effect. And the first day it showed up, one of the high-quality cells, because the cells can be really bad quality, but one of the high-quality cells was showing this just dose-dependent response. And at that point, I was like, I can't even believe this. I don't trust that at all. We have to find every possible reason why that is wrong, because it was just too good to be true.
And what's nice about collaborating together as a journalist, I was in the room when the cell testing was going on, and I knew the players, I knew the science, and so I could record it. My photographer was there. So we recorded the moment this happened, and I was able to take down the dialogue. So when I wrote my story, it wasn't just on, here's the scientific findings. It's here's the end of this quest. I was told it was a story, and so here's there was a tension in the story that built up to the end, and you didn't know as a reader whether this big experiment would work out or not. So I was able to put dialogue in. If you didn't collaborate with a scientist, you wouldn't be there to get those quotes.
Yeah, right. Yeah, it was tense.
Yeah, it was tense. You were nervous.
I was nervous.
So how did you?
That would have been a question for me, because what you described is kind of a classical scientific discovery, right? Which is really, really cool, by the way. I really like it. But okay, that would have been a classical. You have the data at Columbia, you do this observational experiments, and then you do the lab experiments. So what was the role kind of the collaboration of you, Sam, maybe? So why is it a collaboration? Why is it not just an embedded journalism sort of thing, for example?
So okay, two things. One is the approach we take. I wouldn't call too typical. Because of the integrativeness. I mean, if we had just had a hypothesis of ceftriaxone and lansoprazole from some other purpose, then yeah, I think it is pretty traditional. I think the real difference here is the way that the algorithm was shaped as we built it, and the choice and in the direction of the research. And so I would not have explored long QT syndrome or gone down this path at all without the collaboration. And so that's not the only way that it was shaped, but that's the most profound way that it was shaped.
I had got a tip from a lawyer, actually, that there were two popular drugs that were coming together to cause this effect, the effect on the heart. And I liked that story, but it wasn't a great story because the pharmaceutical companies had already changed the label on those two particular drugs. So I said, I don't know if it's really worth spending a lot of time on that. But if these two popular drugs were causing this heart condition, could there be all these other drugs out there causing this heart condition? So that was the big question that I had, the question that we started working on together. And we were talking about before is that was the driving force all along, was this question. We didn't have a lot of bosses to answer to, but we had this question to answer to. That were there other drugs out there that causing this heart condition? Were there 100? Were there a dozen? Could we only prove one? That's what kept the collaboration going. And what I liked about it too is that we all agreed that was the question. Even though you had a bunch of team members and people doing different things, and we talked about different ideas and brainstorming, it all came back. We agreed on that central question.
Yeah.
So how many?
I think you will.
Go ahead.
I just wanted to, apparently, no, this was the journalist's idea for cooperation. Did you have a similar cooperation with journalism before, or is this
really super innovative for you, and would you repeat it?
My collaborations with journalists before this have been more kind of traditional, just reporting results, talking through the results, maybe making comments on other people's research. I had never done, and what I felt was a really equal collaboration before, like this.
Would you repeat?
Yeah, definitely.
Would you be proactive in that search?
I would, I mean, I haven't been, so I guess the answer is no, because I haven't really been, but I would like to be. I think the really powerful thing is that we create a lot of tools. My approach to science is data-driven and systematic. We explore hundreds of thousands of hypotheses simultaneously. So that means some of them are going to be interesting to everyone, and some of them are not. And they're only interesting to people in a particular field, or maybe they're not even interesting at all. And so if somebody can help me identify which subdomain or scope is going to be the most interesting and impactful, then that is a collaboration I would eagerly seek.
So one question concerning this algorithm. So how many drug interactions you pick up with your criterion of these three different measurements? Is it 100 or is it just 2 or was it thousands? And how do you check which to pick?
So in this case, we ended up integrating 2 different data sets. One was the FDA's adverse event reporting system, where we did the original mining, and then the second was the electronic health records from Columbia University, where we looked at the EKGs of these patients. In the first, we mined it, and we generated about 1,000 hypotheses out of probably 200,000 possibilities. In the second one, we evaluated those 1,000 hypotheses, and we reduced it down to 30 to 50 drug interactions. We then rank ordered those by effect size and a little bit of intuition. And then ceftriaxone is the first one on that list that we started. And we have plans and grants to get funding to go down the rest of those, that 20 or 30 that we have left.
My question would actually go to Sam. I mean, how much classical journalism work did you still do? I mean, doing background interviews with, I don't know, doctors, people at, pharmaceutical companies. Like, how much of that classical journalism work did you do?
And for this story, not much. I was working on another story at the same time, something different. But with this story, I think it was sort of contained, that I envisioned this story in the beginning as a story about a scientific quest, that we had this question that was very important to the public, and scientists coming together, and could they answer it, right? And so I wasn't going out and interviewing a lot of other people, because I knew that information probably wouldn't end up in the story, because I had a narrative that I wanted to tell, and that would sort of gum things up. What I wanted to do is tell the story from the beginning, almost like this slide, right? And so early on, I'm thinking like a journalist. I'm trying to think like, how do I write this that people would actually read it? And so when I saw Nick, I saw like a character. He was sort of heroic. So I knew that he would be a main character in the story. And then there was 2 or 3 other people that would be characters in the story. And you have to explain their motivation, and you have to have some tension, and all the things that you really need in a long story. To a piece of journalism, if it's this big, you don't have to worry about character development and tension and protagonist and antagonist. But if a story's this big, you need that, and you need some storytelling. So I was mostly focused on who the people were on the team, where they came from, what their motivations were, and just understanding the science. I mean, we went through this quickly, but to explain it to the average reader, it's complex, right? So you really have to understand it, and then you have to slow it down. In journalism, you have to slow it down and explain very carefully. What's the herd channel? What's this QT prolongation? Why would drugs together harm you? Why would a doctor ever give 2 drugs together would harm you? There's all sorts of things you have to address.
Yesterday you said something about you walked into a pharmacy and bought those 2 medicaments or drugs at the same time. How is this linked to the story?
No,
is it the second story? Or?
No, this is actually sort of different. This is the story I was working on while I was actually working with Nick, a separate story with separate researchers at the University of Arizona and the University of Washington State. We didn't test these 2 drugs, but we did pick drug combinations that would be contraindicated or very dangerous if taken together. And so we identified 5 drugs, 5 drug pairs that were commonly used, and we went into the pharmacies to see if the pharmacies would sell us these drug combinations without warning us. And that took a lot of time and very expensive to do. It was the largest study of its kind, but again, we found that 50% of pharmacies that we tested in the Midwest sold us these drugs without warning us that if you took them together, they could kill you. And because of that, there was a lot of change at these pharmacies across America. The leading pharmacy chains in America, Walmart, CVS, Walgreens, they all made substantial safety changes. And I don't think I could have gotten the universities of Arizona and Washington State able to help me do that story if I hadn't, I think, already been working with Nick, because it gave me some credibility. You know, here's what we're doing here. We're trying to discover drug interactions. So as a journalist, when you start doing these kinds of stories, it helps to just do one, because that allows you to sort of prove yourself when you go to the next scientist. Because scientists are going to be scared of, like, who are you? What are you going to do? How are you going to misrepresent my work? But if you show them that you're serious and you've already done this kind of work, or you're in the process of it, I think they're more open to helping you.
I have 2 questions. One is concerning how did you synchronize? I mean, you have to go to a journal, then there will be a review process, and you don't know exactly, well, at a certain time it will, and you will have pressure to publish it, and you just said, well, I will wait until this is really coming up, and that was possible.
Yeah, it was tricky at the end. The first paper was published in Drug Safety, so I contacted the editor of Drug Safety, the scientific journal, the editor, and said, you know, we would like to run our story. I've never done this before. And I just said, we would like to run our piece of journalism the same day you run the scientific study in your journal. Can we do that? And I was surprised. He said, yeah. And so we sort of coordinated,
Okay.
you know.
Good.
We coordinated, but it's, you know, it adds, in, there was almost like another person coming in.
Yeah.
At one point I complained, I said, oh, I've got so many editors. I got editors on my own paper, editor over here, you know, and they're all like, but it's doable. It's doable.
And the second question is, what happened afterwards? So now you have a drug combination which is not safe. So what happened? Is it still sold, or is there any change in practice, or what happened?
We have since sent a trove of data to the FDA, who have been reviewing it since the publication. The publication that most of the field cares about is this one, and this one was published in November of 2016, I think.
So it's not yet on the label.
It's not yet on the
No.
label, but the FDA has the data and they've been reviewing it. And they did ask us proactively. We told them that we were publishing this. They asked for a bunch of data. We sent it to them, and they're doing their own study right now to decide what to do. In the meantime, we're also sent an application to the NIH to do a much larger scale, and actually to do human trials too. So we'd cover all possible. We'd have the data mining, and we're going to have the cell models, and we're going to couple that with human experiments.
So what did it take for Sam to convince you to work with him? From the scientist standpoint, if a journalist in this room were to come to you like
Yeah.
Sam did, what made that happen?
Well, I think coming out in person from Chicago, I was like, oh. I was like, he really cares about drug interactions. It was really exciting because I'm always like, drug interactions, everyone pay attention. And then somebody was like, I see you're working on drug interactions.
Mm-hmm.
So I was really excited about that. So probably just a little bit of flattery, maybe. But I think just the attitude and the seriousness which with he posed the problem, the question, was the main thing. I'm also generally pretty eager to try new things, so I was kind of excited by that. And in fact, the best story I have about that is that we had been meeting for maybe like, I don't know, a year or half a year or something like that, 6 months. And you had met some of my students. And they came in and they were, after the meeting, and they were like, you could see them like just relax because they were nervous about meeting you. And I was like, guys, why are like that went really well? They're like, yeah, that went really well. He's great.
I don't work for CNN. I mean, I'm not.
Well, then I was like, that went really well, and they were like, yeah, I agree, that was great. We were getting really nervous because it's Sam Rowe, and I was like, okay. And there's like, he's like, he won a Pulitzer, and he's like a finalist for 3 other ones, and I was like, oh, I probably should have looked him up. And that's how I found out about that. Before that, I was just like, oh yeah, journalists who want to work together, let's do it.
I think it helped that we were like-minded. There was a moment where I felt like I was in a good spot when, again, it was like 3-4 months into the project where we were brainstorming and just coming up with ideas, some of them just, unusual, but you were very laid back, and at one point you said, well, I can just have some graduate students work on that. I'll get 3 or 4 graduate students and they can do that and it'll be done. And I said, wow, you can do that? And you said, yeah, let's change the world. And when people say that, and it sounds like a cliche, but it came from you and it sounds so genuine. And I was like, yeah, it was like, great. I mean, this is the kind of, in the newsroom, this is the kind of people you want to work with, people who say that and really truly believe it. And I mean, that was good. And I think that was something that was just exciting. It's exciting to work on a project where, you just have the same goals, and there was no, since we weren't colleagues, there was no professional jealousy. I didn't know much about you, so we weren't strategy. I mean, we just wanted the same thing. It was just the same question. So it was pure. It was fun.
Yeah, yeah.
I need to find more stories like that.
I learned a lot about journalism too, which I really appreciate, and the parallels between the scientific investigation and investigative journalism. We're really after the same goals. We want to figure out the truth as best we can, and I think it's really kind of a natural collaboration. And since then, I've always been surprised that I don't see it a lot more.
Can you tell us again something about the data processing? So for the electronic health record,
sure.
So you have, as a patient, he has certain symptoms,
yeah.
And it's written down in the ECR, and you have access to what exactly? So
essentially every report written and every structured data, piece of structured data that's generated. So in this case, we use the prescription orders, and we use the EKG results, the written results.
For every patient in Colombia?
Yeah, so there was 1.7 million patients in our study and who had EKGs. And then for this particular drug interaction, I think we had on the order of 3,000 to 4,000 patients in that study who were exposed to one, the other, or both of these drugs and had the EKG available during the time that they were exposed.
So once you've had this result in hand, you had to be experiencing a sense of urgency about communicating it because it's a potentially serious public health issue. What was the time lag from that moment of discovery to publication? And implied in this is that there's a lag time for publishing in scientific journals like this one.
Yeah.
And when Sam probably wants to, get it in the paper.
Yeah. Well, you can comment this, but I think you probably wanted to use as strong a language as you could, and then I was probably saying we can't use strong language until we do the cellular test, and so that pushed back. So we had a very short list of interactions, but which one are we going to? That's too many probably to report even in a journal or in a news article, because if there's 30 interactions and they all have these numbers associated with them, and how do you really interpret those? But if you take one and you validate it in a cell model, then you can really focus and you can say here. And I think we ended up with 5 was it 5?
4.
4 that we really focused on, that we thought were the most valuable. And so I think, there was the news result was published before this result, the cellular. So that did get out there faster. We did release all of the cellular data we had at the time as a preprint. So I've been using preprints a lot to get things out faster so that people can start reading them and criticizing them while we wait for the journal.
And you did that in a way that didn't jeopardize the journal's interest in publishing it
Exactly.
after the fact.
We essentially will only publish in journals that allow preprints now. Yeah, there's very few that are allowed that won't.
Do you publish it by archive or?
BioArchive is where we usually put it now.
So what do you think, when will be a label on the trucks?
I don't know. I haven't got an update from the FDA. We should check with Ray to see if he's heard.
Okay.
Ray Woosley, one of our co-authors and a big name in drug safety, especially with QT, this type of arrhythmia. He's kind of been the liaison with the FDA in helping us there.
Okay. And does it satisfy you that it takes so long?
So of course
especially
not.
you.
Mm-hmm.
So if I compare with the Panama Papers, the Panama Papers were published, I think 2 days later, ministers had to deny their office, and your story is much more bigger in the impact and in the essential information, and a year is gone. So as a journalist, I might be annoyed.
Well, there was no competitive pressure working on the story. No one else was going to take this story from us, right?
We're talking about the impact.
The impact, well, we had to be certain. We had to follow it all the way through. I think if you stop the study short or publish before it can be as strong as possible, then you might not get any impact. So in journalism, it's sort of funny. In investigative reporting, we spend a lot of time early in the process working very hard and very fast. And then toward the end of the study, and this end of the project, we were very slow and very deliberate, right? You'd think it'd be the opposite. When you're closer to the deadline, you're racing. It's just the opposite, because you want to make sure everything's correct and right. You want to anticipate challenges, what people might say, because you want to have impact. And all it takes is one error, one mistake. This is what keeps you up at night. All this work, and you make one mistake, there won't be any impact. Because if you're writing about a drug that could potentially kill somebody, the drug companies aren't going to say, yeah, that's my drug. Yeah, it's going to kill someone. Don't buy it. No, and people are going to challenge this, right? And so you want to make sure your story is as strong as possible. So when the cell testing, for instance, pushed the work back months and months, up to a year, in some ways I liked that, because it gave me more time to work on other stories and make sure that everything was correct, because that's the most important thing.
But to this point, I mean, the impact that becomes a little bit out of your control is how long does it take the FDA
Could be
to
years,
approve
could be
How
never.
long does it take the cardiology and medical community to make this part of standard of care, standard practice?
Yeah.
And ultimately, how long does it take, even after you have a black box warning from the FDA on these two drugs? How well does that still get communicated? Back to your other story.
Yeah.
How long does it take for that to be routinely communicated so that it's avoided by the pharmacists and by the doctors? And you have some follow-up stories to do.
Right.
How long it takes the FDA to take these data and
As a
change,
journalist,
right?
as a journalist, if you wait for Congress or an agency to make change, you'll wait forever. And so I think what was great about this project, it was in this cardiac journal here, and so the word got out very quickly, right? And you could have published this in a different scientific journal, but you were held out because you wanted to have it be in a heart journal.
Yeah.
That was more important. And also, as a journalist, the quickest way to get impact on your story is market forces. To write something that forces industry to change. For instance, when we wrote about pharmacies not doing the right thing, not warning us, yes, there was senators calling for action, members of Congress introducing bills, but that may end up with nothing. What really makes change is when companies fear that they're going to lose money because there's these stories out that they somehow are not safe or their service is poor, then they change like that. They end up on their own voluntarily making these changes that so partly they can control the change, but they just don't want to lose money. And so that's what was interesting in writing about pharmaceuticals and things like that, because there's just this fear of bad publicity. And so that's, if you look for market change, you go to the industry and try to get impact, you can get impact quicker that way.
The knowledge of the interaction has spread through the cardiology community, that's for sure. I've gone to some cardiology meetings and they know about the study. And that's happening.
Yeah,
Medicine is slow, stubbornly slow.
But it's interesting though, because cardiologists are not likely to prescribe either of these drugs.
Yeah, that's true. But then they can recognize, but if they're being referred to cardiologists, then they'll be able to identify the effect. I mean, that's the hope, right?
But I actually wonder about the the transfer of knowledge from cardiology literature.
And in order to change like the hospital administration practice, which is going to dictate whether or not what's being used as a first line, for example, then the FDA is going to have to say something, or the insurance companies have to change how they pay.
Are either the pharmaceutical companies you're talking about trying to affect, at the business level, are either of those pharmaceutical companies talking to you about these results?
And that's a good question. So where it stands now is they wanted to see the published results. They've got the published results now, so they were delaying response, and they still say they're studying it. So that's a good point. I should actually call them back. It's like, you'll give us a few more months and call us back because they want to study it themselves. But
I mean, they can actually voluntarily go. I've sat on an FDA advisory panel, not for cardiology drugs, but they could voluntarily come forward to the FDA and say, we would like to add this to the package insert. They could propose it.
yeah, that's definitely something I should follow up on.
That was my question.
But what I like about this drug combination is these are drugs that our readers had heard about. Had we tested drugs that were rare, it might have fallen on deaf ears a bit. But again, these are common antibiotic, a drug that a lot of children take. So I was receiving feedback from readers and questions saying, oh, my child was on that drug, or my husband was on there, what can I do? And so it did resonate with the community. It was a little bit of consumer journalism to this too, which I liked. It helped me sell the story internally at the newspaper. It wasn't just about, it's something, a rare science study. It was something that could affect people. Again, when I could buy that drug across the street at the pharmacy and walk into my editor's office and said, here's what we're talking about, then they saw, oh, okay, now I'm starting to understand. So anytime you have a prop, it really helps.
I get the sense that we were at the time.
Yeah.
All right.
Okay, thank you so much for coming. Really appreciate it.